Hello everybody, good afternoon from Budapest. My name is Marcello Scala and I'm the Director of Sales for EMEA territories for Omixon. First of all, thank you very much for your presence at our annual Omixon webinar. Here with me, I have our guest, Dr. Mette Christiansen, staff scientist at the Department of Clinical Immunology at Aarhus University Hospital in Denmark. Mette has applied NGS for the whole exon sequencing of immunodeficient patients since 2012 and implemented HLA by NGS in routine in March 2016. Thank you, Mette, to be here. I'm also very glad to have here with us Tim Hague, the CEO of Omixon, who is going to give us a live demonstration of the Omixon HLA twin software. Our agenda for today is Mette will present her work on the Biomek 4000, and then Tim will then give you a live demonstration of the latest updated version of the Omixon HLA twin software. And finally, there will be the Q&A session where Mette and of course us will answer your questions, so please wait until the end of the presentation. So now let me give the opportunity to Mette to continue.
So thank you very much, Marcello, and thank you very much all of you for inviting me here to come and present some of our data for the automation of the Holotype HLA on a Biomek 4000. Just to give you an idea of what I'm going to talk about for the next 30 to 40 minutes, the title of the talk said it all in some ways, but what I really wanted to touch is what is the path to increase robustness and productivity by doing automation. I'll give you a little bit of background, why we implemented Holotype HLA, and we have been in routine for a year, so I'll tell you some of the ups and downs from this experience. Which is setting the scene for automation. I'll give you the results from our automation on the Biomek 4000, which we haven't taken into routine. And finally, I'll try to give you some take-home messages. Just to give you an idea of where I come from is we have a center where we both handle organ transplantation and hematopoietic stem cell transplantations, and we have about 300 typings per year of deceased donor transplantations and living donor transplantations. And then furthermore, for the HSCs, we have both typings of patient and family. We have potential unrelated donors. We do about 1,000 typings per year. We are also a center where we have our registered donors, but we don't type them ourselves, but we have quite a few, and we are still considering to do that ourselves. Back in 2014, we had a really complex workflow. We did low resolution by quite a lot of different techniques. We had both serology, we had PCR-SSO, we did conventional PCR-SSP, and a combination of PCR-SSP and SSO. The way to choose what to do was set by which kind of patient, what kind of donor, and for high resolution, we also did a complex mixture of Sanger sequence-based typing. We needed to do reflex testing to resolve ambiguities. Furthermore, we did PCR, SSPs. We sometimes could resolve the ambiguities, and we did additional SPT primers. So this setup took so much time, and we needed to find something different. And as Marcello touched upon, we had been touching the NGS field in the lab, but done nothing at the moment. And then I stumbled on, got some good suggestions from Omixon actually to go to Philadelphia and try out the Holotype HLA. why did we want to do the automation is actually, we have, of course, the goal that we want to increase our throughput, we want to improve the quality, and we want to improve robustness. But before choosing automation, I think it's really important to get the feel of a technique in hand, and we have done so. This process from going through all those different techniques and to a very, very different kind of techniques is a rather difficult task, in the sense that you need to, of course, validate all the new methods you want to implement. To validate them, you need both to train the staff while doing so, and when you're sort of ready, you of course need to have those new methods accredited. At that point, you will often not have trained all the staff that you have in the lab, so there's still some training left before you really go into routine, and this was what happened in our lab. And since then, we've now been in routine for a year, so now we're heading for automation. I'll come by to why, because you can say, how many samples do we run? We run about 24 samples a week. It's not a huge amount, but I still think there are really, really good reasons to automate. Just to give you an overview, I guess a lot of people listening will know what the Holotype HLA is, but just to set the scene for those who are not that much acquainted with it, we're currently using the kit which types 24 samples at 7 loci. And if you go and look at the slide, you can see that the hands-on time at the moment is 5.5 hours. And the total time at this time is 3 to 4 days. Sometime last year we changed from the full flow cell to the microflow cell, which has improved the throughput a lot, and the time from starting up samples to getting out the answers has improved a lot, which has been a great achievement of change for our lab. Because what clinicians in our unit demands are fast answers. The NGS method is based on full-length sequencing of most of the needed genes. I've shown here the HLA, A, B, and C. And compared to Sanger sequencing, what you're usually doing there is just sequencing exon 2, 3, and 4. But when using NGS, it's the full gene. For the DRB1, it's a smaller region that's sequenced, but this is fully enough. And for the QP1, it's the full gene that's sequenced. And what's the purpose of doing full genomic sequencing? I know at the beginning of NGS, and still some labs are doing just exon sequencing, but quite a few problems is connected with just doing sequencing of the exons, because you're not able to phase them, and therefore to get the full potential out of NGS, long-range PCR is the only thing that you can do or should do. If you look at the entrance, for instance, in this case, there is an entering of about 250 base pairs. And if you look at the SNPs in here, you can easily phase them. There are 5 differences between the 2 alleles in intron 2, so exon 2 and 3 can easily be phased with NGS to eliminate genotyping ambiguities. And this is the result of the case for most of the loci and most of the alleles that we look at. But we do have some issues with the P1 typing, where there is a very, very long intron with no SNPs inside, so phasing is not possible for that one. But also going back to when you introduce new things in a lab, you would have to realize that people need to get trained looking at data that are really, really different from what they used to look at. This is not, it's not something that can't be done, but you need to realize that that's something that needs training, especially if you're training people in the NGS technique that has been used to doing a lot of Sanger sequencing. The data from NGS looks so much different that even though it's actually more easy to look at when you're doing it, it poses some difficulties for people who have been used to looking at the peaks from the Sanger sequencing. In my experience, in our experience, people who come with no Sanger sequencing experience will find it very easy to look at NGS data. With Sanger sequencing, a little bit more time is needed, but it will arise, that feeling of the data. So by 2016, I can say now that we achieved our goal. Our goal was to do a setup that was a lot more easy, and where you could say that the goal was to have sort of a very clear idea of what to do when a sample arrives at the lab. So what we do now in the lab is that we have samples that we sort of say, okay, this sample we need to have low resolution typing, or this sample we need to have high resolution typing, but actually we do the same for both kinds. So we do PCR SSP high resolution matching for the low resolution samples, but additionally we in most cases also do NGS. We cannot do that for the deceased donors because deceased donors we need to have results within an hour or so. But for every other organ donation, we really see the force in having NGS as well, because we, by getting the high resolution DP typings, are able to get typings out which enables us to look at the antibodies that patients develop and see whether they are relevant or not. So we always do both typings, and this holds also for the high resolution typing. We do, as the primary technique, we do NGS whenever a sample arrives. And when we do confirmatory typing, we do PCR SSP, high resolution melting. Actually also because when introducing NGS, we sort of very fast found us in a situation where we could do a lot of samples at a time, and we would have very few samples to do PCR SSP on. And since we need to keep people really trained for doing this in acute situations, we need to do both. And I could really recommend this way of typing. So going back to how you can make those radical changes in the lab, and what we saw was that initially, when people saw the technique and looking at the different steps in the Holotype HLA, it sort of seemed difficult. And maybe you could say that to a certain extent, people were thinking, we're so busy, because they were really busy in the lab doing all the Sanger sequencing, the reflex testing, and it was difficult to find the time to really get into a new technique. So they were sort of hesitant in regards to a new technique. And especially a bit later, they would say, okay, we don't really have the time. But when we started and we had got the validation done, we said, okay, we'll try, but it's still taking too much time. But then suddenly things, or suddenly can't say, but it was gradually things changed, and people got really used to it. And one single instance last summer, we had a dropout of the C loci due to some problems in the lab. And they thought, okay, let's do Sanger sequencing of those loci because we know how to do that. But the thing is that it turned out to take so much time and so much reflex testing that it ended up doing, they ended up doing NGS anyway, once again. So they are getting really used to it and seeing that it made that time much better. So at this point, you could say we were ready for automation. But if you look back to the manual Holotype HLA, I could list a lot of do's and don'ts, and I'll try to go and go through some of our data. If you look through the data from March 2016 until November 2016, we used the full flow cell. The problem with the full flow cell is that sequencing takes about 39 hours. So when we started out the procedure on Tuesday, we wouldn't have the results until Monday. This was actually fine, and we could get really good results. As you can see, initially we had somewhere in between 5 and 10% failed loci in the software, but it was actually running pretty smoothly and improving over time. And you can see also that the top part of the graph shows the quality on the MySeq, and that it changed. It was sort of going up and down, and we were sometimes what can you say, worried that things would go wrong, that we were heading in the wrong direction. But it was just people getting used to it, and actually, even though you see those fluctuations in the quality, we got really, really good results. We then changed to the microflow cell in November last year. And this has been a huge thing. It's been so good. We have very, very high consistency in the quality on the MySeq. Almost all the time above 90%. We have very few loci failing, somewhere around 0 to 2-3%. A few are in the last part of the period, as you can see, and that's actually due to some thermocycle problems that we've had, but it's nothing to do with the method in itself. We had to send the thermocycle for repair, so that's what's affecting the loci failing in the last part of the runs. But it's still very few, and for the most part you can say that many of the samples have been typed previously. Either they are for organ donation and we know the typings beforehand, so very few of those failed loci results in repetition of a sample. We actually, most of the time, is able to report everything. And then I would say maybe one sample out of 24 every second week we need to repeat, or something around that. It's very few samples we actually need to repeat. I just wanted to touch upon a thing that we experienced a couple of times, and which actually led us to decrease the concentration loaded on the MySeq, because we had a few examples of very high cluster density, and this is results actually back from the validation, but we've seen it once or twice since And to handle this, we simply decrease the concentration loaded. But as you can see, we have this one sample with a rather high cluster density, and for that same sample, the quality of the data is pretty low. And we, as you could see before, we really don't see that low quality of data any longer. But what we could see is that the fragments, and this graph shows the fragments, the black curve is from a good run, and the blue curve is from the run that I encircled here. And you can see that the fragments are actually quite small, which results in the concentration giving the wrong value, because we think that we hadn't done this fragmentation size selection or fragmentation size testing before afterwards. But so we didn't know that the fragments were rather small, and which was actually due to the fragmentation size selection on the Pripyat prep, resulting in this overload of the mice-seq. So this is a really important thing, but having to do this size selection is pretty neat, but we rarely test the sizes because they are really almost always of the right size. So this is a long list, and I'll try to go through it, but what we've run into is that it's really important to pay attention to efficient amplification. We have a few examples of incomplete amplification due to bad thermocycle, wrong ramping rate, and, as I've underlined, bad PCR plastics. We had some strange results, and changing to better PCR plastics really made a huge difference. And we really rarely see those homozygous calls, wrong homozygous calls now, and we can always trace it back to bad amplification. One thing is that when you do it manually, it's really important to keep focus. That's of course something that holds for everything you do in a lab, but proper preparation for master mix is key to good results, and distractions may be detrimental, especially when you're handling 24 samples at a time. You could easily mess up a lot. This is of course very obvious, but I think when you're getting used to something, you might also get more relaxed in terms of what you're doing, and you would still need to really pay attention. Sample mix-up should be avoided by always keeping the samples in PCR plates and including a known sample in changing positions to be able to really look if you're, I mean, this is needed for FA accreditation, but still it's also a really, really good point to have in there to know that your typing has gone right, and that you haven't changed the position of the samples. We found out that keeping reagents cold and preparing just before use is really important. Shorter fragment sizes increase the degree of cross-mapping and reduce accuracy. And finally, I'd say the library concentration, the determination of that is also really important. It can affect cluster density, and as I showed, too high density results in lower quality data. So tracing samples when you're doing manual pipetting is in the technician's head. So it's really important that the technician is sitting in a nice environment and that there's no distractions, in my opinion, because you're handling all those samples at a time. So going to the automation of the Holotype HLA, why? do you really want to do this? Well, you could say an important factor is less hands-on time. This is important, but in my view, the biggest thing is actually the ability to trace your samples throughout the process. And of course, having a machine do something, you could say that you would achieve high reproducibility as long as you keep your machine good and running, smooth, and doing all the preventive maintenance that is needed. But what does this require? One important thing is to be able to transfer data. Standalone machines will do the pipetting, but you really need to have them on the internet to be able to track samples, or maybe not on the internet, at least on a local network. All processes, in my view, need to be on board as much as possible. So for the library preparation, I would always recommend to have a thermocycler on board. Low requirement for dead volume. Yeah, you can say what you need to look for is a pipetting station that is able to pipette small volumes and not using a lot of excess volume. You can say when you get a kit, you are sort of limited by the content of that kit. And I saw this as a rather big obstacle in the beginning, but it hasn't really turned out to be because of things I'll show you and I'll talk about a little bit more about later. Precision pipetting is, of course, important. You stand there with a low volume of adapters, and if you want to take out those 5 microliters from the bottom, you really need to know that the pipetting station does the trick and it doesn't pipette air instead. And another important thing is to have several pool positions to avoid excess fragmentation going on. So what are the steps in the automation? You can say there is a pre-PCR step where you want to have your DNA normalized and you want to have the amplification set up. Then you have the post-PCR part where you do the normalization of amplicons, the pooling of loci, exocet treatment, fragmentation and repair, adapter ligation pooling. Size selection on pippin prep is, of course, not automated, but you can get ready to do so. And quantification of library is not done on the automated station, but you can get it to pipette the samples to do it. And as I've noted, the really important part in my view is to keep track of samples all the way through this. So I won't go into this slide in detail. It's just to show you at the top. I don't know if you can see the cursor here. Yeah, at the top is the pre-PCR room. So we have DNA purification and pipetting station, the Biomek, and then we transfer the samples to another room where we have the post-Biomek. And I'll come into this more, but what is really important from this slide is all those dotted lines over here, that you can see that all of those, both the pre-Biomek and the post-Biomek and the MySeq is sort of in connection with our server. So we always have the tracing of the samples. So when looking at manual pre-PCR, I won't go into detail, but what you can see is that you have a person sitting there and doing all the pipetting, both when you do the concentration, of course, but we still have that, but also for a normalization of the DNA and pipetting of the samples for the PCR mixtures. And as I told before, you can see that who is keeping track of the samples? It's of course in the technician's head, and then the Holotype HLA worksheet supplied by Omixon, which keeps track of the samples. But these things have to work together, the mind and the worksheet. So the steps in automation, you can say, when we look at pre-PCR, we have to trace sample names, we have to normalize DNA, and set up the amplification. And this is a screenshot from the computer connected to the Biomek. And so we have the sort of method put over here. And why I wanted to show the slide is actually because you can see the setup of this the Biomek station here. And whether you have a Biomek or you order a Biomek, they can be sort of ordered with a specific design. So it might not look exactly like this, but on here you can see there is room for PCR plates, there's room for a cool position for reagents. That's actually here, and there's a room for our DNA samples, and then our room for the tips, pipette tips up here. And so the programming is done here by the software, and then it sort of tells these different tools here what to pipette. And that's, I think, the really neat thing about this station, even though it's a small automation station, that you can put the tools on it that you need. So you can in this instance, you can have single channel or 8-channel pipetting and of the volumes that you need. So when automating the pre-PCR, you can say that we still need the technician in there, because we still have, due to the way our sample comes into the lab, we still do the DNA purification on an EC1. We could do it on another station that would track it in another way, but we get the sample and need to, at the final point, decide what to do. So the technician does the quantification on the qubit, but after that, the samples are put on the Biomek station, and the Biomek station, on the Biomek station, we can scan the barcodes from the samples. We can tell the station what the concentration of the DNA samples is, and we can then afterwards get the station to pipette and do the normalization of the DNA samples in the way we want. After that, of course, then the samples are amplified and results or the sample names are sent to the server. The manual post-PCR is also a sort of a point where you need to concentrate a lot, but using an 8-channel pipette, it's not really a problem. But as I said, it's in the mind of the tech what's going on there. So the first picture is doing the normalization and the first the amplifying concentration, which we do on a fragment analyzer. Then we have the setting up of the library, and finally pooling of the sample and doing the pipping prep, fragmentation, selection, size selection, and then finally concentration analysis. And when this is done, we send it for the sequencing on the MySeq. When we look at the setting up of this, a point I wanted to make was that we are able to do scan of all our PCR plates and thus track which samples are in which position, both on the fragment analyzer, on the Biomek. When we put it in the freezer, we have a barcode and we know that this plate contains which sample in which well. So, and in this programming, we do have different setups. So this first one is for the concentration determination. Then we do dilution, pooling, And finally, the library preparation, pooling of samples is done at the end, and can be done so that we have paused it and we can do the pooling of the samples the next days. One thing I haven't touched much upon is the thing that's situated here, which is the T-robot, which is the onboard thermocycler. This thermocycler, we don't need seals to be put on the plates. We put a pad on the lid of the T-robot, and we replace this each time. It could be clean, but we place it each time, so we can set up this procedure in the afternoon, and it can run during the night time, and we don't have to come and look at it. So once again, you can see here we do have the Biomek. We do the amplicon concentration, we do the size selection, and the results are going to the server all the time. And finally, it sends the worksheet to be used on the MySeq. So what's the advantages of manual or automated? What to do? Well, the automated thing is with low hands-on time. We can do scanning of barcode samples and plates, and we have a worksheet that is generated automatically. One small positive thing about the manual is that there is a small dead volume, but on the automated platform, we compensate this by reducing the total volume, and this works really, really fine, so that we know that we have enough volume there. So what we optimize here is that we fragment the samples either manually or automated, and you can see what we're aiming at is a lot of samples with the fragment sizes between 6 and 1300, and on the automated platform we see actually better results than manually. After pooling and bead selection, this increases a little bit, and this is after pippin prep. We are almost at around 95% of the right sizes. What did we achieve? Yeah, well, we reduced the hands-on time from 5.5 hours to around 2.6 hours. This is really neat, but once again, the neat thing is that we really have sample tracking all the way through. Results are consistent. We have, if you compare results on full flow cell versus micro and the Biomek micro, we actually get very, very consistent results. So finally, I'll give you some take-home messages. Automating this procedure will free some time. But importantly, samples are traced all through the process. Adaptations are necessary, but I would say they're relatively straightforward. We did this in rather few runs on the Biomek. The fragmentation master mix, and this is a really important part, must be prepared just before use, and volumes can be adjusted relatively to ensure that you have enough within the kit. I think I'll stop here and give the word on to.
Thank you very much, Mette, for your time and for sharing your experience from the beginning. I'm sure that there will be a question at the end of the session. Sorry. And now we continue with our live demo of the HLA Twin software represented by the CEO of Omixon team Hague.
Thank you, Marcello, and I'll just echo Marcello's thanks to Mette for that presentation. That was fantastic. The questions are actually open at the moment, so if you have any questions, anything burning you'd like to ask Mette about the automation project that she's undergone, then you can start putting them in now. We should have about 5 minutes at the end to go through some of the questions. Anything we don't have time for, we'll of course send around full email responses to any questions that we don't have time to to get through today. So I'm going to do a quick demonstration of the Omixon HLA Twin software. The focus of this particular demonstration is actually going to be around the new features in the version 2.1 of the software that you can see in front of you here. So if you haven't actually seen the software before, I'm not going to be talking very much about the basic features. You can always contact us at any time if you want to have a more comprehensive demonstration of some of the more basic features of the software. I'm just going to jump in and concentrate on what is new. So this is the screen that you see when you start up HLA Twin, and as you can see here, we have a sample and a result. You can see here a little graphical demonstration of our traffic light system, which tells you that we've got lots of green lights in this sample, a few loci with warnings and no failures according to our own internal quality metrics. So if I go in and view the results, we can start to see some of the new features that we've added into the software already at this level. So at this point, we have a new approval life cycle within the software. So this is to help support the laboratory process and the analysis end of the laboratory process. So you can assign the alleles inside either by manually clicking on a locus. You can see we add some ticks here, or we have a bulk assignment wizard over here as well. So you can actually assign, basically using whichever criteria you like, filtering criteria, you can assign as many alleles for as many samples as you want to on this particular screen. Once you finish with your allele assignments, which are optional but recommended, you can then click on a sample and send it for approval. I'll just show that, and then it says it's ready. So at this point, your lab manager or lab director can come in and they can filter the results and show just the ones that are essentially ready for their attention. And then they can, it's part of their workflow then to approve those results and say that they agree with the genotyping that has been assigned by the lab technician or the bioinformatician. So they can approve these results like this, and it says they're now approved. And at this point, you'll be able to export them from the software. And because everybody logs in to the software, we keep a full audit trail of everybody who does every assignment of an allele, who may override an allele call, and also who has approved this. This can all be logged in the PDF report at the end. So one of the other features that you can see at this level is something called LD up here, which is the linkage disequilibrium check. This was actually added in a previous version of the software, but it's been polished up quite considerably in this version. It's mostly for detecting the hemizygosity of your DRB3, 4, and 5 results. It may also help you to spot if you've had a locus dropout. So if you have a locus dropout, a poor amplification for some reason in HLA-B, it's usually fairly obvious. You have 0 HLA-B results. But for HLA-DRB4, it might be less obvious because this person may or may not have that particular locus. So if there is a DRB4 call that is missing, where we would expect to see one based on the known linkage disequilibrium between DRB4 and DRB1, then we will flag that up inside the software over here and tell you that there's an allele missing that we were expecting to see. And also new in this feature is in this version of the software is a comparison of the DQA1 and DQB1 using the known linkage disequilibrium between these two loci. So if you get a homozygous result, for example, on either locus, this will be checked automatically by the software just to make sure that we haven't had an allele drop out unexpectedly. So it's just a double check on the quality of that result as well. The comments function has also been updated in this version, and as you can see, I checked the HLA-A result and it looked good, but you can put comments at the sample level or the locus level at any point in the software, and the comments are just about in every single screen now. You can still continue to add comments even to a sample that has already been approved, so that's one thing that you can carry on doing as you go through. The last remaining feature on this screen that I just want to highlight is that we have a genotype precision reduction button over here. So if you prefer to see third field results rather than fourth field results, if you'd like to resolve this fourth field ambiguity over here, then you can click on this button and this will all happen automatically. We can also go down to second field results as well. So some labs prefer to run in this mode rather than seeing the full fourth field results that you see here. So if I go into the details of the sample, we can then start to see some of the other new features. So at this level, probably the main feature at this level is that we've added this summary table has now appeared in here as well. So it's easy to see at a glance for your sample what the genotyping calls were and where any comments may be on any of the loci. So you can see that this little red one here is one that has a comment. And if I hover the mouse, it tells me what my comment was. The other main change in here is in the quality control tab. So we've now grouped together our quality control metrics into 3 categories. What we call the primary quality controls. These are the most important ones to make sure that your results are correct. We then have the secondary quality controls. These ones don't often light up, but sometimes can do. And then at the end we have the warnings. These ones generally won't fail, but they may give you some extra informative information if you have had a quality issue somewhere else in one of the other quality metrics. As usual, the most important quality metric of all is probably the read count. If we don't have enough data due to, say, a poor amplification, then we may not be able to give a reliable genotyping result. But that will be clearly visible up here. And every one of these quality controls for every locus has its own set of past info, inspect, investigate, and failed criteria. So every single locus is fine-tuned with these quality control metrics that you see. So most of the rest of the changes that we've made are in the genome browser, so the HLA browser. So if I jump in and have a look at that, we should be able to show you some of the new features in here. So I shall just zoom all the way out so we can see what's going on. In fact, I shall just hide 1 or 2 of these away. There we go. So the first new feature is the one that you can see at the top of the screen. So we have a context-sensitive help panel at the top of the screen up here. It's context-aware help, and it tells you when you move your mouse over it, that when you move your mouse around in the genome browser, it will tell you what you're looking at, basically. This is very useful for people who haven't got a lot of experience with the software. If you're using it for the first time, it may be slightly confusing what you're looking at. And so as you move the mouse around, it will update you and tell you what you're looking at all over the genome browser. And once you've got the hang of it, you can actually turn it off and you don't have to see it anymore. So that's one of the new features in here. We also now have a noise track, so we can display if there's any systematic noise in a sample. There's just a little bit in this sample and in the background over here. So you can see that these are reads that don't really belong to either of the 2 consensus sequences that we've built up here. And so we're not sure where they belong, therefore we've categorized them as some kind of background noise. This can be useful for spotting potential contamination in a sample. If you've got a contaminated sample, you'll see it very clearly in this noise track down here. I'm just going to turn that off again. And another of the new features is to display the amino acids. So, and again here I'm using a display which is only highlighting the differences between the 2 sequences that we're looking at. We've got the consensus sequence up here for the first allele at HLA-A, and the consensus sequence here for the second allele at HLA-A, and all of the identical positions and identical amino acids have been grayed out, and we're just highlighting the ones that are different, both the positions and the amino acids that are different between these 2 sequences. This is a very useful feature if you find a novelty, because you can immediately tell whether or not this is affecting the amino acid coding sequence of the protein. So it's an immediate interpretation of a novelty that is found inside. software. Okay, and I think I've got a nice example. I've got just about enough time for this. So we've got a nice example of a novelty, so I think we'll be able to jump in and have a look at that. So one feature, which is a very old feature, a lot of people don't know exists, which I shall demonstrate, is the bookmark feature. So Mette found out about this today, despite having been using software for over a year. So we have a bookmark feature, and so you can basically take a bookmark of any screen at any point, anywhere in the software, and give it your own name. I've created one called a novel stop codon, so it's probably going to give away a little bit of what the surprise might be for this particular sample. But if I have a look at this one, it's been called as a novel HLA 6603 allele. You can see that over here. And if I look at the coding sequence, where the difference is in exon 3, I can see that I actually have a T substitution. So the reference sequence from the database has a G at this position. The actual consensus sequence for this sample has a T, and that has converted this glutamic acid into a stop codon. So this is a novel null allele with a stop in the middle of exon 3, which I think is a nice demonstration of this new amino acid feature. So that essentially brings to a conclusion the demonstration for today. I realize that if you've not seen the software before, you may have a few more questions about it. Like I say, if you'd like to have a more comprehensive demonstration, then please just contact us and let us know, and we can help you with that pretty much any time. So I'm just going to switch back to the presentation, and we can start to see if we have any questions.
Hi all. So we did have some questions during the demo. We are still welcoming questions. If anybody is having one, please submit your questions in the questions box or on the chat box. So first question is for Mette, I believe. Did you use the 500 or 300 cycle chemistry on the MiSeq for your initial manual work, or did you change it after you switched to using the automated workflow?
Yeah, maybe that wasn't really clear, but the microflow cell is with 300 cycles chemistry, and the full flow cell was used with 500 cycles chemistry. And we used that from March till November last year, and we started doing testing of automation also November last year, but it was totally well, it wasn't related that we changed to the microflow cell at the same time as we started testing automation.
I just received another question actually related to that. What is the throughput of the microflow cell? How many samples do you fit and how many loci in that? What is your normal?
We use 24 samples at 7 loci, and that's really very fine. We get really, really good results from that.
Okay. So I have another question here. It says, how long has the development of the Biomek 4000 method taken? And will it take the same time, or will it be shorter now that it's already developed?
I think it would be much quicker, because I mean, we've gone through all the sort of trials that is needed. So I think, and even given that we haven't spent all our time on it, so it could be done even a lot quicker than what we have done.
So how long did it take originally, would you say?
Well, if in time to spent hours, we've maybe spent a couple of weeks, but it's just been dispersed over quite some time.
Okay. Another question is, do you have any technical documentation to showcase holotype automated on a Biomek 4000? You did show some slides.
I have some slides. I haven't made any sort of real papers or anything, but we are going to.
Okay. So there is another question here. In addition to Beckman, do you have solutions for automation using other liquid handlers from other vendors, such as the Tican? I believe this is a question for Marcello.
Yes, I mean, Omixon can support also the Hamilton Star, the Agilent Bravo, the TPG, the robot DXA from Texas Biogene, QIAgility by QIAGEN, and the Janus Bar by Perkinelli.
And just to add to what Marcello just mentioned, we have already worked with these robots, and we do have scripts for them. But we are happy to work with any vendor, any liquid handling vendor, to collaborate with them to actually develop the method for the particular robot, if that's what is available in the lab. So Tican is also included in that. I have another question here that is similar actually to the throughput that we were talking about earlier. At what sample throughput do you feel that automation was cost feasible?
Well, I must admit we haven't done cost estimations. We felt that this was the right way to go, but I could go and look into it, what the price of the Biomek and the lifetime of the Biomek, but we haven't done that. But I think it's as long as you can find the money, I think it would really improve what you're doing in the lab.
So on a similar matter, actually, there is a question here. It says, I only run 8 samples per run, twice a week. Should I still consider automation?
Well, that's few samples. But we, of course, when we're setting up, we will also be able to do it for 8 samples. It's not just set for 24 samples, but of course, with that low numbers, it would probably be better to do it by hand.
So another one, just the last one for now. It has to do with sample number. So the question is, if we reduce the sample number and use a full flow cell, can we expect better results, or maybe it's similar to microflow cell?
Yeah, it's a funny question though, because the full flow cell accommodates up to 100 samples. So if you reduce the numbers, it doesn't really add anything because there's so much data in there but few cells works fine as well. But it's just giving you much more than needed. And 8 samples on a microflow cell is really, really fun.
Okay, and just one last question, because we are running out of time here. Can you handle single locus testing with the 11 locus holotype testing?
Sorry, could you just...
Can you handle the single locus testing?
Oh, yeah.
For example, the monotype HLA with the 11 locus holotype testing?
Easily doable. We haven't set up our automation for that because we don't want to do that at the moment, but it would easily be done.
So just to add to what Mette just said, yes, it's definitely doable, and a lot of different robots are able to handle that, exactly because they are able to cherry pick what needs to be done for each one of them. So it does improve the consistency and the accuracy of doing that process. So we do have a couple more questions, but we have run out of time, so we will make sure to respond to everybody by email.
So thank you, Effie, thank you, Mette, as well. And I think before saying goodbye, I would like to introduce in the next slide my Holotype page. My Holotype page is an authenticated area of our website, where basically we provide tools and resources to facilitate the use of our product, such as manuals, such as workbooks, and also data set to be more familiar with the HLA Twin software. Well, saying that, I think I want to thank you everybody. Again, I want to thank you, Mette, for their presence here in Budapest. I want to thank you, team, for the and all the staff from Omixon. And obviously, I want to thank you for the participation at this webinar, and I wish you have a good rest of the day.